So class, in this recording, we're going to continue our discussion of the genome, the human genome, and how we pass our genetics on to the next generation. As we look at this process of passing our genetics on to the next generation, from going from parent to child, it's useful to look at a karyotype. And when we talk about a karyotype, we're talking about a chart that has 46 squares on it, or rectangles on it. And this grid represents the 23 chromosome pairs. So we have 23 rectangles here. And as we look at these 23 rectangles, we can have a duplicate one for the sex pairing. So as we look at 1 through 22, that represents the autosomes, and then that 23rd pair could either be an XX for female or an XY for male, generally speaking. So if we were to look at, just to make it really clear, let's say if we were to look at a female's karyotype, the female's karyotype would not have that XY region. So it would look like this and have 23 slots filled. And if we were to look at the male's karyotype, instead of having two X chromosomes, generally speaking, all males are going to have an X and a Y chromosome. And that's what their 23 slots would look like. Now, as we look at those 23 pairs, just to review, we get one pair from mom, one pair for dad. The first 22 pairs within the karyotype are the autosomes, and they look alike and carry the same genes, and we automatically are going to get them the same autosomes from mom and dad. With, accommodations made for individual alleles, different alleles within the same kind of gene. As we look at those sex chromosomes, we have one pair of sex chromosomes. Females are going to be XX. They'll have the same pair, so they'll be homologous. They'll have two of the same sex chromosome. Males tend to be heterozygous in that they will have an X and a Y sex chromosome. And this is a good rule of thumb. XX for females, XY for males. We'll have a separate discussion on the biological determination of gender, where we talk about SRY determining regions and some of the weird exceptions like XXY or XYY, so on and so forth. But for a rule of thumb, XY is going to be male, and then XX is going to be female. Now, as we look at the human genome, our genome is diploid in that most of the cells of our body have two sets, 2N, of chromosomes. So any cell that has 23 pairs, or two sets of 23, is going to be diploid. This is most of the cells of the human body. However, reproductive cells are haploid, and these haploid cells have only one set of chromosomes. And as the two kinds of haploid cells that humans have are sperm cells or egg cells. These combined are generally speaking, are called the germline cells. And these germline cells, or germinal cells, are important. We need to have haploid cells for reproduction. Otherwise, if we reproduced with diploid cells, we would continually double the number of chromosomes with each subsequent round of human reproduction. And that's not a good thing, because we have lots of diseases associated with having too many chromosomes. So to keep the number of chromosomes stable in the human population from generation to generation, well, we make these sex cells. We'll start with a diploid cell, turn it into a haploid cell, and then reproduce with the haploid cells. And when those 1N haploid cells are combined together, that's called fertilization, and we go from haploid back to diploid. This process of making the sperm or the egg is referred to as meiosis. And just to review again, that sperm or egg can be called a germ cell. Oftentimes, though, it's also going to be called a gamete. And we start off with that parental chromosome. And that parental chromosome, or that parental cell, is going to duplicate its chromosomes so that we have a chromosome from dad and a chromosome from mom. And then those chromosomes from dad and chromosomes from mom are ultimately going to be separated so that we have daughter cells that have only one set of DNA. So instead of having two chromosomes from one from mom, one from dad, the daughter cells would have one chromosome, either one from mom or one from dad. And we go through that we repeat that process for each of the 23 sets. Every once in a while during this process, we're going to have crossing over. One part of a maternal chromosome and a paternal chromosome are going to flip-flop during this process of making a gamete, during meiosis. Crossing over typically is going to happen at the very beginning of meiosis. So let's clear the screen a little bit. Let's look over here. The parental chromosome, the male chromosome, it's colored blue with a green region on it. And if there's no crossing over, the gametes are going to have blue chromosomes with a green region on them. However, if there is crossing over, like there is on this side, look at this. We have one paternal chromosome with the green gene, and then another paternal chromosome that has the red gene. So one of the genes from mom's chromosome got added and flip-flopped with the genes from dad's chromosome. And this crossing over event that occurs during meiosis adds a lot of genetic variation to us as a species because we have sexual reproduction. And it's one of the big advantages of sexual reproduction. We tend to roll the dice with our genetics a lot more. Now, as we look at a specific gene, our favorite gene, whatever AFG, our favorite gene is, it's going to be at a locus on a chromosome. And that particular locus is going to describe where to find that gene. And you can have different variations of our favorite gene. Those different variations are referred to as alleles. And typically, we're going to have a dominant allele and potentially recessive allele for individual traits. When we're looking at dominant alleles, we're usually going to abbreviate them with a capital letter. And as we look at recessive alleles, we typically abbreviate them with lowercase letters. So let's talk about a good example, dimples. There's an allele that controls whether or not you have dimples on your cheek when you smile. And because dimple starts with a D, I would call the dominant allele for that a capital D and the lowercase allele, the recessive allele, a lowercase d. So the letter of the alphabet chosen usually is going to correspond to the first letter of the trait associated with that allele, and then we have the capital for dominant, lowercase for D. As we look at the dominant allele, that dominant allele is almost always going to cover up the effects of the recessive allele. So if somebody inherits one dominant and one recessive allele from mom and dad, only the dominant allele trait is typically going to be displayed. The only way we can see a recessive allele's trait is if an individual inherits a recessive allele from mom and a recessive allele from dad. So they need to be homologous for that specific recessive allele. And these recessive alleles are usually, not always, but usually going to code for some non-functional variant. So a lot of our genetically based diseases are going to be recessive allele-based diseases. Not all, but a lot. So as we look at genotypes, our genotype is a way of describing the individual alleles that an individual has for a specific trait. And they have two of the same alleles. Oops, let's go back here. If they have two of the same alleles, we call them homozygous. You could be homozygous dominant and have two dominant alleles, one from mom, one from dad. You could be homozygous recessive and have a recessive allele from mom and a recessive allele from dad. Or you could be a heterozygote and have one recessive and one dominant from either mom or dad, and you can flip-flop that. So when we talk about genotypes and we're saying homozygous, you have to say homozygous dominant or homozygous recessive most of the time. It's really not necessary to say heterozygous for a dominant and recessive allele. Just by saying heterozygous for the allele, it's implied that it's for the dominant and recessive allele. Now, as we look at phenotypes, that's going to be whether or not the trait has been displayed. So genotypes are the genetics of that specific, the allele that the individual inherited for a specific trait. Phenotype is if the trait is expressed. Usually, we're going to see the dominant phenotypes expressed. So cleft chin, here's a picture of John Travolta, he has a cleft chin. Homozygous dominance individuals or heterozygous individuals will express the cleft chin. And then we have the uncleft chin. Uncleft chins are going to be on individuals that are homozygous recessive. And if we take someone with John Travolta's genetics and someone with Arnold Schwarzenegger's genetics and have them reproduce, we can make a Punnett square to show the potential probability of an individual child having an individual genotype and phenotype. So let's say dad has John Travolta's genotype. So one sperm cell, one gamete could have a dominant allele, one could have a recessive allele. And then as we look at the mother has Arnold's genotype. So she has a dominant allele, and actually they both have, I shouldn't say Arnold's genotype because mom has a dominant and a recessive. So both mom and dad are going to be heterozygotes in this particular example, Punnett square. So dad's a heterozygote, mom's a heterozygote, and if two heterozygotes reproduce for a trait, there's only a 25% chance that the recessive trait is going to be expressed within an individual offspring. And then a 75% chance that the dominant trait will be expressed in an individual offspring, assuming simple dominant and recessive genetics. That's all we have for this recording. The remaining recordings for this lecture are going to focus on a lot of Punnett squares and interesting genetic situations. So stay tuned for more. And as always, happy studies.